All right. Okay, I've added a drop of undiluted sperm, and you can see that it's pretty wild right here. There are a ridiculous number of sperm attempting to get through the jelly coat that surrounds this egg. There you can see the sperm zipping around with considerable vim and vigor. They've even got little tails and all that. And I deliberately over-fertilized this one. In the wild, there wouldn't be nearly this many sperm per egg. But here's an egg. And this one has been fertilized. It happened really fast, as you might expect. And there's a whole bunch of sperm that are trying to push through that fertilization membrane, but they can't do it because of the slow block to polyspermy. This one has also been fertilized. So has this one. And in fact, pretty much all of the ones on the slide got fertilized before I could even start the recording. There's one. Maybe there's some up here that didn't quite get clobbered. Okay. Yeah, here's one that hasn't been fertilized yet. And you can see there's a bunch of sperm around there. They're trying to push through that jelly coat that this egg has. And as soon as one of them gets through, the fast block to polyspermy is a wave of electrical polarization that spreads from the point of fertilization all the way around the egg. And it happens in a fraction of a second, but it only lasts about a minute. The second thing that'll happen is a layer of very small vesicles that are right now right underneath the plasma membrane of the egg will release substances out of the plasma membrane. They release, I believe it's proteins that absorb a great deal of water, and they'll take the fertilization membrane that surrounds the egg and physically lift it off the surface of the egg and form a barrier that those sperm can't cross. So we're going to see if we can see that happening. When it happens, it'll happen as a little bump appearing along the side of the egg. And that bump will get bigger, and you'll see the fertilization membrane lifting off and spreading around. And if anybody's curious, this discovery of two different blocks to polyspermy, the fast block and the slow block, was made by a scientist named Ernest Everett Just. Just had the misfortune of being a cell biologist before the molecular revolution. He did his best with light microscopes, not that much better than what I've got right now. But he got his PhD in 1905 and died in, I believe, the 1940s, before the discovery of the DNA double helix, before the widespread use of transmission electron microscopy and a whole bunch of techniques like that. And yeah, he had like a lot of scientists at the time, he's partly forgotten now, just because he lived at a time when it wasn't possible to do so many of the things that we take for granted now. But armed with just a microscope, he was able to work that out. And he did a lot of pioneering studies on embryos of marine critters at Woods Hole Oceanographic Institute in Massachusetts. A lot of very solid basic work in developmental and cell biology has been done in marine laboratories, and you can see why. It's easy to collect eggs and sperm. You can do lots of experiments in bulk. There's lots of different things that you can do. Just, by the way, is also partly fallen into obscurity for another reason. He was, in fact, African American and worked at a time when certainly a lot of people didn't want to believe that an African American could be doing science, to say nothing of, could be doing cutting edge cell biology. Even the people who backed him, his monetary supporters and people like that, could be very condescending and suggest that maybe he should be doing something a little more practical with his time. He taught for many years as the head of the biology department at Howard University. I believe that's the oldest and one of the most prestigious historically Black colleges in the United States. Ended his career in Europe, where he could pursue research without the pressure of racial discrimination that he had to face in the United States. And in some ways, his work got left behind simply because, as I've said, there's so many more things we can do and experiments we can run and questions we can ask and answer with molecular biology techniques that were simply not conceivable when Just was working. However, in some ways, he was ahead of his time because he was famed for his incredible experimental technique. He would show up at Woods Hole and he would always know exactly what time of night, what night of the year, the urchins or the parchment worms or his other critters might be the most fertile. And he would observe the eggs under very carefully controlled conditions that were as close to nature as he could possibly get them. There were a number of his colleagues at the time were trying to work out developmental processes by watching development and then zapping the eggs with some kind of toxin and seeing what that did. It's a little bit like the particle physics approach, where you try to study a watch by smashing two and examining the pieces. And Just argued that that really doesn't tell you anything. You can make embryos do all kinds of weird things by treating them to unnatural conditions. But if you really want to know how eggs develop and how embryos develop from fertilized egg to adult, you've got to look at the way they interact with their environment. You've got to look at the way that they interact with the place that they have adapted to live in. And only then can you really say anything about how development goes. And one of the things that we have learned is that he was right in that embryos don't develop in some kind of environmental vacuum. They don't develop separate from the environment that they're in. And understanding that environment can give you insights into developmental biology. Understanding how the environment controls development is a field that we're only now starting to look at, the field of environmental developmental biology, or ecological developmental biology. And Ernest E. Just was a pioneer in that field, and some of the things that he realized and wrote about in a book of his called Biology of the Cell Surface are the sorts of things that we're only now coming to appreciate, that we're realizing are much more timely than perhaps we used to. So I'm going to continue to watch this egg and see if we can get the fertilization membrane rising. I'm not sure why it hasn't happened yet. But I'm going to go ahead and leave the camera on it. The others that surround it have all shown a membrane. So, here's one with a very clear fertilization membrane. The sperm look like they're starting to realize they're not getting a date tonight. And there's one. That membrane probably started appearing at about the 5 o'clock because it hasn't lit. It's lifted the farthest off the egg there. And there's another one that has. And there's the only one that so far has not. Okay, I'm still seeing a few sperm that are trying to break in. So I'm going to leave this on for just a little bit longer and see if we can see the fertilization membrane rise. If we haven't, at least you now see what it looks like. And then I'm going to look at this slide again in about 1 hour and see what we can see of the first division of this embryo. So I'm going to give this one just a little bit more time, and we will see what we can spot. I'm moving around just a little bit, looking for some more. Yeah, there's one that's been fertilized. There we go. All right, so there's the one where we're just not yet seeing that that fertilization membrane. Yeah, when you add this many sperm, fertilization happens so fast that it can actually be rather hard to catch it before to catch it on video before it's happened. I'm wondering if that egg might not be defective in some way because all of the ones around it. Sorry, moving. Yeah, like that one right there. Yeah, that's been fertilized there. Yeah, that black dot that I'm seeing at about the 10 o'clock position, I'm not sure if that's the sperm nucleus or not. The egg is packed so full of yolk that it can be hard to see details on the inside, although you can see a slightly lighter spot in the middle. That's the nucleus. All right, and this one has still not quite been fertilized yet, but at least you got to see what the fertilization membranes look like. And I'm going to turn this off and look at it again in an hour, and we'll see what the first cell division looks like, because an hour is about as long as it will take.